William, and Josh were two persons linked with a murder scene. At the site of the murder, police collected a weapon as well as some hair strands, which were handed over to the forensic team and were asked to find out the actual murderer by performing some tests. But the murderer was very clever and he underwent plastic surgery. So when the results of skin fingerprinting they came out, none of them matched for that. The team was now confused. The only option left now was of DNA fingerprinting, as it cannot be altered by plastic surgery. It was found that the murderer was Josh, who did the plastic surgery just to escape from skin fingerprinting. So, what is DNA fingerprinting? Just like skin fingerprinting, this can distinguish one human being from another with the exception of monozygotic twins. While skin fingerprinting, it can be altered through plastic surgery just like Josh did, but DNA fingerprinting cannot be altered by any means. It remains same in every cell, tissue and organ of a person. So, this is a structure of DNA containing two helices and nucleotides. The nucleotides, namely adenine, thymine, cytosine and guanine, which are linked together by multiple bonds. DNA fingerprinting is a technique used for determining these nucleotide sequences of certain areas of DNA which are unique to each individual. This is really surprising to know that there are only 0.1% of genome which is different in each individual and which makes every individual unique. Rest other 99.9% of genes, they are similar in every individual. These differences they occur not only in genes but also in repetitive DNA such as in these two chromosomes. Here we can see that these repetitive DNA have a unique sequence. These are known as satellite DNA because in density gradient centrifugation, in which components of cells they are separated on the basis of a density, when placed in a centrifugating machine it forms a small peak, while bulk of DNA it forms a larger peak. So one of such repetitive DNA is variable number tandem repeats known as VNTR, which has about 11 to 60 base pairs which are surrounded by conserved restriction site. These differ in families because of some small deletions, insertions and mutations. And how do these occur? Because every child receives about 50% of DNA from father parent and 50% from mother parent. Therefore, the number of VNTRs in a particular area of two homologous chromosomes, they are likely to be different. As a result, each individual comes to have a distinct combination of VNTRs which are peculiar to that person only and not to anyone else, except in case of monozygotic twins. Now let's study about technique of DNA fingerprinting. First of all, we need to find a source of DNA which can be anything like red blood cells, hair follicles from the root, white blood cells, semen, saliva, vaginal swabs or skin cells, etc. Then we have to extract the DNA by using high-speed refrigerated ultracentrifugation which will separate the nuclei and break the same to free DNA. These DNA they are then amplified by a technique known as PCR, that is polymerase chain reaction. The DNA is then fragmented with the help of site recognizing restriction enzymes and DNA is then cut to separate VNTRs. This chopped DNA is exposed to electrophoresis over agarose polymer gel, which separates the DNA fragments. The separated VNTRs they are recognized by staining them with dye that becomes fluorescent when illuminated with UV radiations. The separated segments they are also known as RFLP, that is restriction fragment length polymorphism. Those separated VNTRs they are then transferred to a nitrocellulose or nylon membrane which is placed over a gel, which is known as Southern blotting. DNA probes, they are then added to this nitrocellulose or nylon membrane and which get attached to single-stranded VNTRs having complementary nucleotide sequence. They are then exposed to X-ray films. The hybridized radioactive VNTRs, they then appear as dark bands like this, and then the film gives us DNA prints or DNA profiles. The prints, they are then compared and relationship is found. It can be used in identification of criminals involved in various types of crime including rape and murder. It can also be used to provide reliable information as to real biological father, mother or offspring. It can be used to establish the closeness of relation of an intending immigrant, or it can be used as an information for human lineage and relationship with various apes. Isn't it interesting? So that's all we have about DNA fingerprinting. In case of any doubts, you can comment it down below, or until then, see you. Thank you.